Go ahead and get started. Welcome, everyone. Thank you for being here today. Our speaker today is Dr. Jeffrey Spraggins. Dr. Spraggins is an associate professor in the Department of Cell and Developmental Biology, director of the Biomolecular Multimodal Imaging Center, and director of the Mass Spectrometry Research Center at Vanderbilt University. He received his BA in chemistry from the College of Worcester and his PhD in analytical chemistry from the University of Delaware. He is a world leader in spatial omics and spatial biology. His research group is known for one, developing novel mass spec techniques to maximize imaging performance, enabling molecular histology at the cellular level, and two, engineering multimodal workflows that combine imaging mass spec with a variety of other biomedical imaging technologies to provide a systems-wide biology view of the tissue at cellular resolution. The Spraggins group applies these techniques to the construction of molecular atlases as a part of the NIH Human Biomolecular Atlas Program, the NIDDK Kidney Precision Medicine Project, KPMP, and the NCI Human Tumor Atlas Network. Welcome, Dr. Spraggins. Thank you for being here today. And for all participants, please feel free to write your comments in the chat during Dr. Spraggins' talk, or unmute and ask the questions directly at the end of the talk. Thanks. Take it away, Dr. Spraggins.
Thank you very much for that kind introduction. As was just said, my name is Jeff Spraggins, and I'm here at Vanderbilt University. And we develop technologies around molecular imaging and apply these to a wide range of applications. But one of our primary focuses over the past 10 years has been applying and developing these approaches specifically for the study of the human kidney. And so when we think about multimodal workflows and molecular imaging, we're really, developing tools that allow us to bring together all of the technologies that you see here. And so all of these approaches are instruments and technologies that we're developing in-house and have here at the Mass Spectrometry Research Center at Vanderbilt University. And so we focus on everything from kind of untargeted, label-free forms of microscopy, simple things like autofluorescence microscopy, as shown here on the left, where we get, kind of broad scale morphology of the tissue. It's low specificity, but it's cheap and it's relatively high resolution. And so you'll see examples in the talk of where we're leveraging things like autofluorescence to segment features of the tissue and use these modalities to help us mine our deeper molecular assays, things like imaging mass spectrometry and spatial transcriptomics. In addition to unlabeled approaches, you'll see examples of where we're developing strategies and applying technologies for highly multiplexed immunofluorescence microscopy. There's various tools that are emerging to do this. We use a cyclic-based approach, something called codex or co-detection by indexing. But in all of these strategies, effectively, you can build up panels of tens of protein antibody targets so that you can quantify the organization of the cells within the tissue and relate that to other modalities. What we're kind of most known for is imaging mass spectrometry, and I'll spend kind of the first portion of this talk just describing this technology in some detail and explaining how it works. But effectively, we're using the mass spectrometer to generate molecular images. So it's kind of combining the molecular characteristics and specificity of mass spectrometry with the spatial components and spatial fidelity of traditional microscopy. What's nice about this is we're not using any probes or targets. We're measuring the endogenous molecules directly from the tissue. And so if you can detect those molecules in a mass spectrometer, then we can typically apply this approach to your sample. And so we take thin tissue sections, we can look at metabolites, lipids, glycans, proteins, we can look at post-translational modifications. Typically, we're detecting hundreds of molecules in a single experiment. And although it's high molecular specificity, on the spatial side, we tend to be a little bit coarser than in microscopy. So we tend to be in the area of about 5-micron pixel sizes. So I like to say it's cellular resolution, but not single cell resolution. And then we take these untargeted molecular profiles and we can relate them to other modalities, things like spatial transcriptomics or spatial proteomics, to get at kind of mechanistic information and pathway level information within these samples. As was also mentioned, we apply these as part of a number of large-scale consortia. This has allowed us to kind of engineer solutions to perform these intricate technologies at scale across human samples. And so we kind of started down this path as part of the Human Biomolecular Atlas Project, or HubMat. The goal here was to develop molecular atlases for quote unquote normal human tissues. And our first grant with this program was to develop an atlas of the human kidney. And you'll see some of the initial work for that program here in this presentation. We then followed up with another grant as part of HubMap to look at the human eye, and the human pancreas. But we're also part of the Kidney Precision Medicine Project, a program that many of you might be aware of that's funded through the NIDDK, focused on chronic kidney disease and acute kidney injury, as well as the Accelerating Medicines Partnership, AMP, looking at autoimmune disorders. And then our most recent grant is part of the Human Tumor Atlas Network, where we've partnered with Ken Lau here at Vanderbilt, who's the director of the Center for Computational Systems Biology. And there we're focused on colorectal cancer. And so the technologies that we're developing are kind of central to our research group, and then we can apply it to a whole host of application areas. But in general, what we're really focused on is utilizing these multimodal strategies to define whether it's normal aging or disease in molecular terms, so that we can identify biomarkers for improved diagnostics, and eventually, partner with folks to utilize these targets as starting points for novel therapies.